I'm James, and I started my PhD here at the University of Surrey in 2023 in January. So that makes me something like 2.5 years into my project now, and it's definitely ramping up. I've only got a year left, so it's really crunch time. The doctoral college match fund my PhD with the Prostate project, and my PhD is investigating the ethnic disparity in prostate cancer. So we've collected 114 patient samples from 3 centres across the UK, the North Bristol NHS Trust, the Royal Marsden and the Royal Surrey. And within this cohort we have a mixture of Asian, Black and white men with prostate cancer. And the project is to try and understand the biological reasons which could underpin ethnic disparity. 1 in 8 white men get prostate cancer, whereas there's 1 in 4 black men get prostate cancer. And within that statistic, black men have a 60% higher risk of succumbing to disease compared to white men. So there's quite a drastic disparity. So the first really big experiment of the project is something called shotgun metagenomic sequencing. And this technique allows you to profile the microorganisms which live within your body. So normally in something called symbiosis, or in a healthy relationship, these microorganisms act in a beneficial way, which coincides with normal physiology. However, in pathophysiology, such as cancer, there is outgrowth of nasty bacteria in something called dysbiosis. And this has been linked with multiple different diseases, not just cancer. But the idea is that by profiling the microorganisms within the prostate tumour itself, we can start to understand whether there's unique signatures of these bacteria which may correlate with a worse outcome, or maybe found within different ethnicities. And we've started to actually get this data back and perform analysis, and we can see that there's an enrichment of certain bacteria within the tumour compartment. The second big experiment is that we're profiling the tumour microenvironment of these tissues. So we have 114 prostate tissues, and what we do is we investigate the 3D tumour biology on a 2D plane by investigating the different cell types and how they interact with the tumour. We can then start to understand what types of therapy could be an option for these patients, and try and understand really the complex biology that underpins the tumour, and by doing this it can inform decisions. So currently all of the wet lab experiments on the bench have finished, and currently I'm performing a lot of analysis. So spending a lot of time in the dry lab, performing both the analysis for the microbiome study and the tumour immune microenvironment, both of which take a lot of time and take up a lot of data storage, as I'm sure you can imagine. But we're getting a bit closer, and hopefully I can have it all wrapped up by my end date, which is June 2026. Thank you to the Prostate Project and Univar for your continued support. Without you, I wouldn't be able to do this research.